Yes, thank you for the introduction. So today I'll be presenting a method that we've developed during the last 2 years of my PhD, which I completed indeed in Utrecht, the Netherlands. So let's start with at the beginning and let me explain why we would even want to find shared SNPs. So let's consider as an example these two traits, type 2 diabetes and rheumatoid arthritis. And if we look at the SNPs that are associated to these traits, we would find some SNPs that are associated just with type 2 diabetes, and these SNPs might be associated with insulin signaling, for instance. And for rheumatoid arthritis, we might find some SNPs that are involved in joint inflammation. But we might also find SNPs that are associated with both traits. And in this case, in this example, these SNPs could very well be involved in the immune system because these are both immune-related traits. And these shared SNPs are also called pleiotropic SNPs. But why would we look for them? So if we can identify these SNPs, we can find traits that have similar underlying processes, as I illustrated here in this example. We could also potentially find out more about the role of a SNP that had an unknown function previously. And if we would zoom in on just one trait, we can understand better which underlying processes of this trait are specific and which processes are shared also with other traits. But how would we do this from GWAS summary statistics? So first I want to say that it would be really nice if we can do this with GWAS summary statistics because they can be publicly shared. They're not privacy sensitive anymore. And nowadays we have an abundance of them available in public databases. So again, for this example, for these two traits, we can, for each SNP, which is a dot in the plot, we can plot the effect size of trait X on the x-axis and of the other traits, trait Y on the y-axis. And then if we would go back to these SNPs from the previous slides, we would find the SNPs that are involved in joint inflammation on the y-axis because they have an effect on rheumatoid arthritis or trait Y, and they do not have an effect on trait X, and vice versa for insulin signaling. But the SNPs that are involved in the immune system, in my example, they would be found in this corner, or actually one of the corners, because they have an effect on both traits. Yeah, and so before we can actually move on, I first want to clarify that there are actually two types of pleiotropy. So in the first example, horizontal pleiotropy is where a SNP, the red bar on the bottom, has a direct effect. So this is not an association, but a direct effect on two traits, on trait X and Y in this example. Vertical pleiotropy is when one SNP has a direct effect on one of the traits, but because there is correlation between the traits, it will also show an association with trait Y. And since we can only find associations, if we don't do anything, we would find both types of pleiotropy, but we only want to find the horizontal direct pleiotropy. And so, because this vertical pleiotropy is caused by correlation between the traits, we want to do a decorrelation transform. And then we see that also, or particularly the SNPs in the middle, the gray SNPs, that don't actually have an effect on any of the traits, they also show correlation in the upper example. But if we do this decorrelation step, it is gone. So now we can actually move on, and any pleiotropy we will find will be this direct horizontal pleiotropy. And now let's actually talk about what defines a shared SNP. How will we find it? So first we need to realize that we want to find SNPs with an effect at all. And for this we use a distance from the origin as a measure. And But a SNP with a large distance can still have a trait-specific or shared effect. And so we also want to look at the angle with the x-axis, because that tells us whether it's trait-specific on one of the axes or whether it's shared. And these metrics are actually the polar coordinates, so we can readily convert this from Cartesian coordinates. And then actually, like I said already, it's not only in that one corner that we would find pleiotropy, it can be in any corner. It depends on the original direction of effect, but each corner is equally shared. And so to make things a bit easier, we fold the quadrants. And then to help the further analysis, we still want to make sure that these angles describe a full circle. And so we multiply them by 4. So now all the pleiotropic SNPs will have a transformed angle of pi, or 180 degrees. And all the trait-specific SNPs will have an angle of 0. So now we again need to take one step back and think about, okay, what is actually a large distance? What is large enough? So remember, the purpose of this step is to get rid of the SNPs that just don't have an effect at all, not trait-specific, not shared. And a SNP that would have an effect has a distance that is larger than expected for a multivariate normal distribution. So we can just readily use a central chi distribution with, in this case, 2 degrees of freedom for the number of traits. And after this, we have gotten rid of all these SNPs in the middle, and we continue with only the filtered SNPs that have an overall effect. And now we are going to define sharedness using this angle, the transformed angle. And the purpose of this step is to get rid of the trait-specific SNPs. So we will only have the shared SNPs. And here we already saw that all the shared SNPs have an angle that is close to pi. And for this, we use the von Mises distribution, which you might not have heard of, but it's similar to a normal distribution, but then for angles. So it takes into account that an angle of 0 and 2 pi is the same, for instance. And after this, we can actually identify pleiotropic SNPs. And I did that for 41 traits. So these are all pairwise applications of polymorphism. So for every pair of traits that you see in this network, I ran polymorphism, and I'm showing here the width of the edges here shows how many genetic loci are shared by 2 traits. And there's really already a lot we can learn from this network. But one of the most striking features is probably that, oh, and by the way, I colored it on disease domain. So one of the most striking things here is that the blood pressure domain is very strongly connected. And remember that I decorrelated the data. So this is really independent from genetic correlation between traits, where you could make a similar network, of course. This is really telling us how many individual genetic loci are shared by each trait, regardless of their correlation. And another very closely connected domain here is the lipid traits. I don't know if you can see my mouse. No. The light blue one, so total glycerides, TG, total cholesterol, TC, and LDL and HDL. And I want to zoom in a bit more on that domain. Because, yeah, like I said, it struck us that this domain was so connected. And so I was wondering, have we actually found all SNPs that are shared by all these traits? So before, I've always been talking about finding shared SNPs between 2 traits, but now we would want to find SNPs that are shared by more than 2 traits. So first, again, I want to walk you through the motivation for this. If we would analyze these 4 traits of that example in a pairwise manner, we would have to do 6 individual analyses and then take the intersect of all the shared SNPs. And then we would know which SNPs are shared by all 4 traits. But on the other hand, if there would be a way to analyze these traits together and find that answer immediately, this would entail doing just one analysis. And now actually the number of analyses you'd have to do scales very quickly with p, the number of traits. But for a joint analysis, it would always be 1. So this was really clearly a motivation for us to figure out how can we apply this for more than 2 traits. And now I will walk you through the example for 3 dimensions, just to keep it a bit easy. But actually this way of this approach works for an infinite number of dimensions. So here I've tried to draw a 3-dimensional plot. And we see, wait, maybe I can use the mouse. And there it is. So we see 1 dimension per trait again, trait X, Y, and Z. And the dotted line indicates the trait-specific effects. And we're considering this red SNP here, which is also described by the vector X from the origin to the SNP's position. And we want something similar as the case for 2 traits. So first we want to know the distance. This is actually a very straightforward extension of the distance in 2 dimensions, so we can do that. But then for the angle, we need to first think about, okay, what is the null hypothesis? It's still that an effect is trait specific, just like the case for 2 dimensions. And so a trait-specific SNP would lie on one of the axes. And to keep it a bit simple, we first just want to find the closest axis. Well, that in itself is relatively easy, because the closest axis is the one with the highest value, so Z in this case. And now we've redefined the problem to what is actually the angle between the observed SNP vector, the red X1, and the null hypothesis vector, the mu vector, the yellow one. So this angle. And fortunately, the angle between 2 vectors is always in a 2D plane, no matter the number of dimensions we're considering. So this really makes the analysis much easier. And we can find that angle by realizing that the dot product between 2 vectors is the product of their distances times the cosine of the angle between them. We can rewrite this, and we can rewrite this again by realizing that the dot product is also the sum of product of its terms, and the square of the distance is the sum of the squares of its terms. So now we can calculate both the distance and the angle in the multidimensional case. And now we have found a solution for an infinite number of dimensions. Now I do want to share the results as well of this joint analysis, of course, because we already saw that it would theoretically be more efficient. You just have to do less analyses. But we found that it's also more powerful. Because again, the same example, we found 186 individual genetic loci by doing this pairwise analysis and taking the intersect. But for the joint analysis of these 4 traits together, we found 1,029 genetic loci. And I forgot to put it in the slide, but almost all 186 loci of the first example were also found in the second example. So this is a tremendous increase in power. And now I hope that I've managed to share some of my enthusiasm for this method. So PolarMorphism is a relatively efficient method to identify pleiotropic SNPs, which works on a theoretically infinite number of dimensions. Of course, your computer might disagree, but it does work on GWAS summary statistics, which is nice because they are not privacy-sensitive data anymore. And it uses parametric distributions. So there's no need to simulate null distributions, and this is one of the reasons that it's relatively fast. The code can be found on GitHub, or our results can be found on Zenodo, a European public database. And it is also published. So I will show you the QR code again on the next slide. And I'm just really hoping that some of you will pick this up and apply it to your favorite trait combinations, and then hopefully find new biology. And now I just want to thank my former colleagues, my former lab, and specifically also Michelle, a bachelor student who helped me quite a lot with the literature study for this project, and Jeroen and Sander, my previous supervisors, and René Eikemans from our biostatistics department for just going through the statistics with me and making sure it sort of makes sense. And thank you for listening. And there's the QR code.
There is some time for questions.
Hello. That was very interesting. Thank you. I was just curious, when you're taking the higher dimensional angle, you're taking it essentially the smallest angle to an axis, right? So that's a kind of extreme value choice that you're making. And I was just wondering if the same null distribution that you would use when you only have one choice would be the same that you would use when you pick an extreme value.
Yeah, that's a very good question. Thank you. I'm now realizing I forgot to say something, and I also left something out on purpose. So the thing I forgot to say is that we do have to correct this angle that we find, because it is, as you are, I think, implying, it is always bounded by some maximal value. If it gets larger than that, another axis will be closer. And so that's why we calculated a correction factor. Well, we have an expression for the correction factor that depends on the number of traits P. But also, this von Mises distribution has two parameters. A mean, which we set to 0 for the null hypothesis. The angle should be 0 for trait-specific effect. And also another parameter, which is sort of the inverse of the variance. And that also depends on the number of traits P. No, I'm making a mistake. It depends on R. But we show a complete derivation of this in the paper as well. But yeah, that's definitely a very important thing.
Yeah, I've got a quick question. If I understood you correctly, that your study is mostly focusing on the SNPs. I was wondering, would you be considering to look into the copy number variations as well as the indels? Because a lot of traits have a lot to do with the expression levels as well as the copy numbers.
Yeah, thank you for that question. I've been thinking a lot about extending this method, because I think just the basic principle of this would probably be applicable to a lot of different data types. As far as I can think of, all it needs is just some effect size or some significance estimation on the same entity. Whether these same entities are SNPs or indels or copy number variation, I don't think that would matter. Yeah, I think in principle it really should be applicable to that data as well, as long as you have some effect size for the same CNV for different traits or different outcomes. Or
okay. So you showed a more than 5 times increase in effect size in your power when you're doing your joint analysis.
This one, yeah.
Compared to the taking the intersect. Where do you think that effect comes from?
Yeah, good question. Of course, I've wondered this myself, and I recalculated this a few times because I didn't believe it. I think the main power increase is coming from the fact that we have to correct, as in all GWAS things, we have to correct for multiple testing. And if you do 6 different analyses, you basically have to correct for multiple testing 6 times. I think that's where the main power increase is coming from. Yeah, I hope that answers your question.